So now that we've gone through the process of meiosis one, we've replicated our DNA in interphase, and then we have those tetrads form and cross over to give us unique genetic combinations. And then we had independent assortment occur to give us even more variation. Then we had those cells split up, and we turned one cell into two. Now we have meiosis two, which is finishing off the process. And now here, I don't want you to get confused, but meiosis two is very similar to mitosis, or at least the mitotic phase, where you just have prophase, interphase, metaphase, anaphase, and telophase. Okay, but we'll talk about it. And I want you to remember, these are very different processes. Okay, but meiosis 2, if you know mitosis, you pretty much have meiosis 2 down. So no interphase 2, or interphase 2 is very, very short. There's no DNA replication going on here. And remember, meiosis 2 is similar looking to mitosis. So prophase 2, not particularly exciting, but chromosomes condense, the spindle forms. Prophase 2 is actually a bit of a snooze fest, to be honest. So I probably won't ask you many questions about prophase 2. You should just kind of know that it's there. Metaphase 2 is the same thing as metaphase in mitosis. Chromosomes are lining up on the metaphase plate. They're lining up at the equator. Remember, in metaphase 1, you had that tetrad at the equator. You had both homologous chromosomes that had replicated itself. Now we are looking at chromosomes lining up at the equator. Anaphase 2, those chromatids are getting separated into the poles, very much like mitosis, very much like anaphase of mitosis. And then ultimately we have telophase 2. Okay, the nuclei reform, cytokinesis occurs where they're pinched off from one another, and you wind up with 4 haploid daughter cells, as they're called, at the very end. Okay? And again, these are gametes. These are sperm, and these are eggs. In fact, if you want a little bit of extra credit, write me an email that says gamete extra credit, and then in that, I want you to tell me what is the ploidy, or are gametes haploid or diploid of a gamete, and I want you to tell me what is the ploidy. Again, is it haploid or diploid of just like a random skin cell? Okay, and you should also, tell me what that means. So you should say something like, "Oh, gametes are haploid, meaning they have this many chromosomes," and or whatever your answer happens to be. So at the end of telophase 2, you wind up with 4 cells. Okay, 4 genetically distinct cells. So let's go through a couple of questions. If you have a cell with 20 chromosomes, so this is not a human cell, obviously, it only has 20 chromosomes, at the beginning of meiosis, at its completion, how many chromosomes would you have? Okay, so diploid, you have 20 chromosomes. How many do you have after you undergo meiosis? So again, think about, what are we doing in meiosis? What's the point? We are going from a diploid cell to a haploid gamete. We're reducing our chromosome number by half. So if we start out with 20, at the end, we should have 10 chromosomes in a haploid gamete cell. I know this is all brand new language, but please stick with it. This stuff is important. This stuff is useful. The next concept I want to talk about is this idea of a karyotype, and they used to do things called a karyotype splat, where they would just take a few cells and they would sort of splatter them onto a slide, and then you were able to actually look at and figure out which chromosome is which. This kind of has to happen when a cell is is beyond prophase, because you have to be able to figure out which chromosomes are which. And if they're all uncondensed, well, it doesn't make any sense to do one of these. But here you can actually pick out the chromosomes. What they used to do is they would take a picture and then they would actually physically cut out each chromosome and then line them up next to each other. These are arranged by size. And I also want you to notice that you have 1 through 22, which are autosomes. And then you have this last pair over here, which are your sex chromosomes. Autosomes are different from sex chromosomes, just so you guys are aware. If you are XY, you are a male. If you are XX, you are a female. We're going to talk about those details later on in this semester. But you should know you have 22 autosomes and 1 set of sex chromosomes. So here's another picture of our karyotype. And here, yeah, this is a picture of a female karyotype because there are 2 X chromosomes. Now, in the next section, we're going to talk about different genetic anomalies. I think I'm going to save this for next week, but you can see right here, this is Down syndrome. This is a person with trisomy 21, meaning that they have 3 of this 21st chromosome here. And again, next week, we're going to talk about how that can take place and what sort of issues that can cause.